is Evan Wiederstedt. I'm a computational biologist with the Department of Biomedical Informatics at Harvard Medical School. I also have affiliations with the Broad Institute and Massachusetts General Hospital. Today I'd like to walk through what we've been building over the past several years with the cell annotation platform. Before I jump to the site and walk through some of the features we've built, I wanted to give a conceptual grounding about what this infrastructure really is and how it helps us solve some of the problems in the field at the moment. The background to all of this is, of course, the Human Cell Atlas, whereby we're trying to come up with comprehensive characterizations of cell types in the human body, trustworthy biomedical references that will be used to spawn research directions that we really can't even fathom. This is very similar to what took place within the Human Genome Project, whereby we had human genomic references of DNA. In this case, we're looking at the transcriptomes, the RNA, of individual cell types with unlimited precision to see what is their identity and their function based on the genes that are expressed. Now, a key little sticky part of the data analysis here with single-cell genomics is that after scientists process the data sets and they cluster together cell types, they will associate these clusters, you can see on the right, let's say green, orange, red, purple, blue clusters, these clusters with a biological entity. And they do this looking at gene expression of the transcriptomes. Now, the problem that has always sort of existed in this field is that there's really not a shared vocabulary for how to do this. An example on the left is that for new cell types, which logically we don't have a vocabulary for the new cell types we've never really characterized before within single-cell genomics, scientists will simply provide cell annotations in a paper with maybe some numbers, some abbreviations, luminal cell 1, 2, 3. And this is the best way at the time they can communicate these results to their colleagues. On the right, you'll see there's also a question here of how broad and how precise should these cell annotations be when communicating results. And normally this is based on the expertise of the biologist. A biologist with an expertise in the immune system will provide far more detailed, precise cell annotations than, let's say, a biologist who is studying a system that she or he may not know. And of course, the elephant in the room, and the reason why this is really so complicated, is that because we don't have these references, we don't have these reference atlases which tell us the truth of how do we define these cell types, we have to rely on really just biological knowledge. And there's healthy debate that takes place across the globe regarding how do we define these cell types? What do we use for, let's say, key marker genes, that are the genes used to define these cell types, for systems either within broad fields of study or across disciplines. An example of the former is that there's healthy debate within the immunology community about how do we define, let's say, development states of T cells. In the latter case, there's connections between these biological systems. Let's say the retina community will deal with the nervous system, and the nervous system community may have some key ways that they define these cell types that are simply unknown to retina researchers. So that is the key motivation behind the cell annotation platform, which I think boils down to two main problems. One is one of scientific communication and community standards. How do we encode these cell annotations such that scientists can effectively communicate with one another? How can we encode these cell annotations such that the data can be useful to achieve the scientific results that we're trying to achieve? The second problem here is really one of, it relates to how do we really create these biomedical reference atlases for the Human Cell Atlas? And the first problem is simply, what did the cell annotations mean? How were they created? What do the labels mean? Are the labels clearly associated with the data, such that scientists could go back and investigate whether they trust these cell annotations or not? Who created these cell annotations, and when did they create them? We've tried to solve this problem by constructing a metadata schema to ingest the data of cell annotations that prompt users to provide more information about what do the cell annotations mean. That is there known terms that exist in biomedical databases or ontologies? Are there synonyms that exist for the term? What are the marker genes that were used to define those cell annotations? This is useful for scientists to see whether this is true in the data when they actually do the data analysis themselves. It's a reproducibility issue. And secondly is some rationale to communicate with other scientists in a far more effective way than scientific publications about what did the cell annotations mean and how were they generated. The second problem is the key goal of the Human Cell Atlas. We're providing trustworthy biomedical reference atlases of healthy cell types and cell states for the community to use for their downstream analyses. How do we plan to do this? The key word is consensus. We need both consensus in terms of how cell types are defined, but we also need consensus in terms of whether the cell annotations associated with the data are believable or not. And this is precisely what CAP has provided. With easy-to-use UIs, researchers will investigate the molecular data and they'll be able to provide public reviews, something akin to, let's say, Google restaurant reviews, in terms of whether they believe the cell annotations of their colleagues. We ask them to agree, disagree with some rationale, which is structured for downstream analysis, or say they're really not quite sure. The two workflows I mentioned is one, we need consensus in terms of the cell type, so we ask users if they agree with the biological definitions provided for the cell types, but we also ask users to work with the data itself and agree with or disagree with whether they believe the cell annotations connected to the data are correct or need modification. This was a slide shown at the HCA Asia conference in 2023 in Kolkata, West Bengal. It's a key illustration of how the Human Cell Atlas is organizing the community to systematically review the cell annotations of a reference atlas. By doing this, we can collect where there's community agreement and where there's community disagreement, which needs to be revisited. By timestamping and versioning this, we'll be able to provide to the community really the best possible and most transparent way to really record whether cell annotations are believable or not. It will be the largest references to date, and it will have the opinions of the top biologists in the field recorded, whether they trust these cell annotations or not. After several rounds of the cell annotation jamborees, we'll be able to timestamp these and release these for the community. With more data modalities, we'll be, of course, refining these cell annotations, which is another reason why they are timestamped. And this is roughly a conceptual overview of how CAP is so valuable for the scientific analyses. In the next video, I'll walk through the features that we use to accomplish these goals. Thank you.